Okay. Can you see the screen?
Yes, sir.
Okay. So we were able to have that lectures last week because of the exams for other batches. So I decided to have 1 hour today, and then maybe during the week we'll have another one. But tomorrow we'll have usual time. Okay, 9 isn't it, right? We have at 9, right?
Yes, sir.
Tomorrow. Okay. So if you remember, right, we were talking about the history of rice growth improvement, rice varietal improvement, right? And then we were the last class we studied like briefly, right, some basic understanding about growth stages. But we will go into detail in the coming classes to growth stages. Today we are going to have a detailed look on this progress, right, of rice growth, rice improvement program in Sri Lanka, right? So in the meantime, right, if you have any problem, like if you cannot hear me, please let me know. Okay. So this progress of rice improvement program, you can look into step by step, right? Because several important events occurred to develop rice varieties in Sri Lanka. So we can look before 1930s, right? Some history, what has happened before this time, and then after that, right? What we were doing is pure line selection, right? From traditional varieties. So in any genetic improvement, the selection is the very important procedure which starts at early, right? So you will have obviously so many varieties, right, for crops, but you need to select the varieties with good characters, high yield, maybe resistant to diseases, pests, ability to survive drought or something like that. So by looking those characters, you obviously select some varieties. So that was happened the second step. So from the traditional varieties, now you have studied what are the traditional varieties we had, what are the problems we had with those traditional varieties. And then the third step was introduction, right? They were not happy with these traditional varieties even after selection. So they try to introduce some varieties from other countries, right? So then they thought they can bring preferable characters to the rice genetics in the country, right? And then once they bring these different varieties, the variety development started, right, through hybridization and selection process, right? Even after hybridization, we have to select, right? Select the varieties, right? So hybridization and selection. So that was the fourth step. Is the major one through which we have developed these old improved varieties and newly improved varieties. So I have explained them, right, in the previous class. What is old improved variety and what is newly improved variety, right? And then we will, in the next tomorrow's class, this the fifth bullet point, right? We will have a look and look at briefly what are the future varietal improvement strategies they are working on, the Rice Research and Development Institute working on, and then what are the prospects, right? So if you look before 1930s, right? Before 1930s, so there were little work reported. You may know, right? Before independence, we were ruled by the British, right? The last, last people rulers were British, right? So the rice sector was neglected by these people, like British people, because they were mostly focusing on these plantation crops like tea, rubber, and etc. Right? So the rice sector was neglected, but during the World War II, right, importance of rice as a staple crop was realized by the rulers. Why? Because this with the World War II, right? There were a lot of problems, especially in importing, right, rice. Because earlier they were importing and they were focusing on plantation crops. But now, because of this World War II, right, that has affected so many countries, right, during 1940s, right. So, so there were problem in transportation and other stuff, right, economic issues and all. So they were realized better to produce or better to improve the local production of rice, right. So in this interest intensified in local rice production in late 1930s, right? So in alignment with this, this World War II and this increasing interest, so the history says that there were investigations on rice production and rice varieties were carried out by those rulers just before this independence in 1948, right? So there were investigation. They were working on, what are the areas rice is produced, what is the extent, and what are the varieties that are good to produce some yield and all. So they were doing some investigations, right? So that what happened as the initial step, right? So then, so they started working on pure line selection, right? So in the last classes, I was talking about we had many traditional varieties, but they had so many inferior characters. But even though within those traditional varieties, they were trying to select the lines. Lines mean you can consider as variety or maybe so different types of rice, right? So they were trying to select those pure lines, right? From those traditional varieties. So this Paddy Advisory Board was established in 1943, right? So this was the sole body to advise the government on the matters related to rice production, right? So they were established in 1943. So the traditional varieties were purified for yield stability, right? So the pure line means, once you select that variety, you can continue cultivation. It doesn't change its characters. So if you provide required management, your yield will be stable, right? So they were looking for that, right? We had so many traditional varieties. From them, right, they selected these pure lines, right? Pure lines, especially considering the yield stability, right? Whether they can maintain the yield throughout the season, right? So here I put some of these isolated varieties and their duration. So Murungakayan, like Kodivi, right? Pachapemal, right? There are different numbers and they are line numbers. And you don't need to worry about that, right? That BP28724, like, and then this Sudhihinati and Halhinati, those are the varieties selected, isolated from the traditional varieties, right? You can see, right? The duration also ranging from like 3.5 to 4.5 months, but not less than 3.5, right? So not less than 3.5 is still more than 3.5, right? But this Pachaperumal is one of the popular pure line selection, but it was susceptible to blast disease, right? That was the problem, right? It's still problem they had, but they were able to select, isolate, right, some lines for yield stability, right? So that what happened in this second step. That is the pure line selection from the traditional varieties, right? Are there any questions so far? Students, do you have any questions in these two steps? Okay, so if I put a question like what has happened like in the first step, that's before 1930s and the pure line selection, can you explain? Yes or no, please tell me. Otherwise, so if you don't talk, I will not continue. Okay, I will stop and go. Because this is important. This question often comes right regarding the history and what, right? So that's about the pure line selection, right? So let's move to the third, right? Kind of milestone, right? So that's introduction from the other countries, right? So I said that we had still problem with this pure lines, right? Some of them are like susceptible to diseases, and we had still like the long duration varieties. Most of them are like 4 or above that, right? And so we were unable to grow like 2 seasons, right? So then they thought, let's bring some varieties from other countries. So the problems with traditional varieties were recognized. So major problems, I already taught you, right? What are the problems, right? So many problems, but among them, so these are the major problems with these traditional varieties. The lodging, right? Because they grow tall, right? They grow tall, so the lodging means this they fall down, right? With the wind or maybe when they near the harvesting and all. And this is susceptibility and poor nitrogen fertilizer response, and the lower yield, right? Because of all these problems, they had lower yield, right? So these are the some of the varieties introduced from other countries to Sri Lanka, right? PTV 16 from India, right? And Mas from Indonesia, and Sigaris from Indonesia, right? So there are some other varieties as well, but these are the three popular ones introduced to Sri Lanka from other countries, mainly from India and Indonesia. So these varieties had moderate response to nitrogen fertilizer, right? Nitrogen fertilizer. So this is the step involved in introducing new varieties. If you are introducing a new variety, right, that means maybe in agriculture biology you may have studied, or maybe you will study. You are bringing a new gene, right? A set of genes, right? So new variety means new genes, right, into the country, right? So after this introduction, they decided to work on hybridization and selection, because these are like foreign varieties, and they were also not much performing well to the situation in our country, right? To the agroclimatological conditions, they were not performing well, right? So then they were thought we have to work on hybridization and selection, right? So you may have studied this also in the agriculture biology, or otherwise you will study. When you develop a new variety, the process usually we use there are new varietal development methods in the genetics available, but it's kind of old. But fundamental method is hybridization and selection. So you need parents, right? That means two cultivar, right? You mix them then you will get a F1, right? You may have studied in biology advanced, right? So that's the first generation, right? F1, right? And then this F1 will be multiplied several times, right? Until um, we get a pure variety. So for that, you have to go for like 7 to 8 generations. 7 to 8 generations. So first step is that you select parents, like two, right? Two varieties, and then you breed them. So that's hybridization. After that, you will get F1, right? F1, that F1 multiplied several times. Like we call it as generations, right? So when you reach usually F7 or F8, right, seventh or eighth generation, you will get a purified variety, right? So that takes a lot of time. Maybe like you have to wait at least 7 8 years, right? 7 8 years, or maybe if you have 2 seasons, then you can bring it down. But generally, it takes like more than 5 years. Okay. So this hybridization and selection process to improve rice varieties in Sri Lanka occurred in 2 phases. So you can guess why 2 phases, because you know that we had old improved varieties, OIV, and then newly improved varieties. So both of them are produced through this hybridization and selection process. Okay. So this national hybridization program started in late 1950s in Sri Lanka. So initially at the agriculture station located in Mahailapallama, then this work shifted to Batalawada, considering these irrigation facilities. You may know, like we have a field crop station now, Field Crop Research and Development Institute in Mahailapallama, that was earlier rice research. Rice research also conducted, started there, but that location had minimum irrigation facilities. But for rice, as you know, a lot of water is required, so irrigation facilities required. So they thought to shift this location to Batalagoda. So now the Rice Research and Development Institute is located in Batalagoda with good climate conditions and irrigation facilities. Okay. So as I explained, right, they followed this hybridization and selection process. So this first step, right, in the phase 1, so they developed the H series. So this H series were called as old improved varieties. Okay, old improved varieties. This occurred between 1958 and 1969, right? So this is the time period, the phase 1 of the hybridization and selection in rice happened, right? Where they developed this old improved variety. So we call it as H series because it has an H, and then the variety names with capital H and then numbers with some numbers. Right, that's why it's called as H series. So this H indicates the hybridization, right? Hybridization. Earlier they named like that, right? So in this phase 1, H4 was the first bred variety, right? So through this breeding, right, the first old improved variety was H4, right? H4. So let's look at this first old improved variety H4, right? Pedigree or parents are the Murunga Kayan. So that was the one of the traditional variety, right? Selected the line from the traditional variety. You can see here, right? So we have an example as isolated pure line, right? So that one of the parent, and then mass, right? Mass is the introduced one, right? Introduced one. So you can see here, introduced from Indonesia, right? Introduced from Indonesia. So those are the parents, right? For this H4, right? H4. So this variety released in 1958, right? So that's why, to release a variety, like you have to spend 7 8 years. So that's why I said They have started working on, started in 1950s, somewhere 1950s. An average yield was 3.5 tons per hectare, and maturity is around 136 days. It's around 4.5 months, right? Still 4.5 months, right? It was still sensitive to photo period, right? But it had moderate resistance to blast, disease, and moderate response to added nitrogen fertilizers, right? So because of these beneficial characters, the yield improved over the pure lines, right? Before this hybridization and selection, we had pure lines, right? So those are the lines selected from the traditional varieties. But with this improvement of old improved varieties, that's the first one, H4, right? The yield has improved over the pure lines, right? So here in this table, once they developed the first old improved varieties, they were developed some other old improved varieties as well. So in this first column, you have some of the old improved varieties, H4, H7, H8, H10, H105, H501, right? And these are the old improved varieties released by the Department of Agriculture. You have the corresponding released year, right? And then duration, and then their major characters. You can see most of them, right, old improved varieties were red pericarp, right, varieties, right? Red rice, not the white one, right? Then only, right, when they started working on the newly improved, only they were giving priority to the white rice varieties, right? White rice varieties because the majority of the population, preferred those white rice varieties. But still, if you go to some areas, maybe in the northern region, Jaffna or maybe Kilimanjaro, in the northern province, and also in the deep south, if you go to Matara, right, they still like the red rice varieties, right? Some of them, right? But more, if you have to remember the old improved varieties, majority of them were red, right? Red, right? Only we have H8, that's the Samba, right? Right one. But majority of them were red. But you also can see their characters, like they were blast resistant. H4 was resistant, and then H7 was resistant. Again, H10, H105, they were resistant to blast because that was the disease mainly affecting those days, right? So they were trying to incorporate those characters in this development, right? Are there any questions so far regarding this first phase of this called improved varieties before we move to the phase 2?
So why they poor response to the nitrogen fertilizer?
You mean which variety? So which variety you asking?
The traditional variety.
Yeah, traditional variety. Okay, traditional variety. As I said, right. So maybe I can go back, right? So see this slide.
Yes, sir.
Yeah, they are taller, right? They are taller, and then their leaves, when they grow, right, their leaves are droopy, right? So they don't do much photosynthesis, right? Much photosynthesis, and they are, if you look their leaves, right, their chlorophyll content and everything was less, right? So when they do little photosynthesis, right, what will happen is, they don't demand, especially the nitrogen fertilizer, right? Because they have little chlorophyll content, and then they are taller, they cannot absorb the light. So they, when the demand is less, right, then they will not uptake, right? They will not uptake the nutrient. So that's why they had low response to this nitrogen fertilizer. Is it clear?
Yes, sir.
Yeah, because whenever we don't feel hungry, right, we don't eat much, right? It's like that, right? When they demanded less, right, they don't uptake. So that's why, with this hybridization and selection, all these improvement, we were trying to develop a short variety that can absorb a lot of light, right? A lot of light, and then with a high chlorophyll content, so then they can do a lot of photosynthesis. Then obviously their demand is high. Then they will be respond to the added external nitrogen fertilizer. Any other questions? Others? Okay, so I hope so you are now clear with this first phase of hybridization and selection, right? So in the first phase, right, we have developed the old improved variety. So there are some milestones you should know, right? So H4 was the first old improved variety, right? And then these are the characters that particular old improved variety had. And then you should also know most of the old improved varieties were America varieties, right? And then the phase two, right? In the phase two of hybridization and selection, this genetic procedure involved in this process was same for phase one and phase two was same, but the varieties used for that process was different. That's only different, right? Because the techniques involved, especially in the breeding, was same, right? Just hybridization and selection. You have to take two varieties, they are the parents, and then you will get F1, right? F1, and then using the F1, you multiply and taking so many generations, or you may stop by F7 or F8. So then your variety will be having stabilized characters. Stabilized characters. So usually it takes like 7-8 years, right? So in the phase two, we have developed these newly improved varieties. So because of this problem with these taller varieties, we use this dwarf, right? Dwarf means short, right? Short varieties. So if you are using a variety, means you are taking the gene from that particular variety, right? So they were high response to nitrogen fertilizer with high tillering, right? High tillering. You know what is tillers, right? So these pictures, right? See, so the rice as a grass, it will has a main stem. Like we don't call it as a stem; it's a culm, right? And around that, we have so many small, right? Stems coming up. So we call it as tillers, right? Tillers. So it's a one single plant. Here you see, but it has, around that main culm, it has so many tillers, right? Tillers. Each and every tiller can produce the panicum, right? Where the grains are produced. Okay. So high response to nitrogen fertilizer with high tillering, because they are short varieties, they can increase their photosynthesis, so they can demand more nitrogen, right? And then a range of maturity classes from 2.5, right, 2.5 month to 5 and 6 month varieties were developed, but most of the varieties around like 3 and 3.5 months, right? These newly improved varieties. So in this phase two, right, in developing newly improved varieties, we have incorporated resistant to major pests and diseases. Like if you take pests, right? So these are the major pests considered to incorporate the resistance, like BPH, that's a brown plant hopper, and then rice gall midge, right? That was also a problematic pest, especially in the Asian countries. And then diseases like rice blast, bacterial leaf blight, BLB, right? So the genes responsible for the resistance to these pests and diseases were incorporated. and also the tolerance to abiotic stresses like salinity, stress, iron toxicity. So this iron toxicity was one of the issues, especially in the wet zone rice cultivation, especially in Kalutara area, right? Iron toxic soils were there, right? Especially in the lowland, right? So we had to, right? Salinity problem observed, especially in the coastal areas where rice is cultivated. So we had to incorporate those tolerance genes into this breeding program, right? Breeding program. So how this happened in Sri Lanka is that in parallel to the development, right, green revolution began in Asia, right? So this green revolution, you should know, right, started in wheat, right, because that was the major crop in the world. But in the Asia, right, when you come to Asian countries, the rice was the important crop, right? So they brought that technology to Asia, right? So this green revolution in rice improvement started in 1949. So especially in two places. One is in Taiwan, right? One is in Taiwan, and the second one is in the International Rice Research Institute. Can anyone know, right? Tell me where this is located in International Rice Research Institute?
Philippines.
Where's that? No, I am talking about Asia, right?
Philippines.
Yeah, Philippines, right? So International Rice Research Institute is located in the capital city of Philippines, that's in Manila, right? So in two places, right, in the Asia, this green revolution started, right, especially on rice. And these are the two parents, right, they used in Taiwan, right? I am talking about Taiwan. So the first one was, this was the common one. This is the very popular one, DGO Wugen. Sometimes they abbreviate as DGWG, right? Because this particular rice variety had a character, that short character, right? Because we said the major difference we made is we brought the height to smaller height, right? Because the earlier varieties were taller, we had to have, right, the short varieties, right? Short varieties. So this is the variety had that gene, right? The dwarf gene. Dwarf means short, right? So this is the dwarf rice mutant, right? DGOVUGen. Sometimes they call as DGWG, right? So that was the one first parent, right? Second one was this one. They are own one, Taiwan one, Kasai Yanshun, right? So that has it was told, but it has some disease resistance, right? So they want to incorporate that disease resistant to this dwarf short variety, right? So then they use these two parents to breed this particular variety in they used as parent was PETA, right? PETA, it was told, but it had high T11 ability, high T11. So they wanted to incorporate that. So they used the PETA, right? So using these two crossing, right, and they have bred this variety IR8. So this is um, one of the popular first improved variety in Asia, right? In Asia, IR8, in Tamil also they call IR8, right? I really studying this one because it's happened somewhere in other countries, but we used it, right? That's why I want to explain happened in Thailand and Philippines, and then in Sri Lanka, right? So let's move to how they developed NIVs in Sri Lanka, right? So they earlier, right, they want to test this TN1 because we had, poor breeding facilities and all, right? So they thought, the rice scientists in Sri Lanka, they thought like just bring this TN1, right, TN1 and IR8, right, to Sri Lanka and test them, right? So the TN1 and IR8 were tested in Sri Lanka in the late 1960s and early 70s, right? Early 70s. But they failed, right? They failed due to their susceptibility to diseases, especially we had a problem of bacterial leaf blight. These two varieties were not resistant to this bacterial leaf blight, abbreviated as BLB, right? And then they had, this in the they required this intensive crop management, right? So our farmers were not that much familiar with this intensive crop management earlier, right? Now they are familiar, but earlier it was new to them. So because of those reasons, this TN1 and IR8 introduced to Sri Lanka and tested in many locations in Sri Lanka, but they were not performing well. So what the Sri Lankan rice breeders thought was like take a semi-dwarf variety called Enkatek, right? So this is an Indonesia variety, right? A gene from Indonesia, right? To develop these newly improved varieties, right? So that's why I explain briefly this, this history of this one. TN1, yes, we use TN1 and IR8. We try to test them, right? But they were failed. But another semi-dwarf variety using same principles, like we used was what we were using was Enkatek, right? Indonesian variety. So using this particular variety as a parent, right? As a parent, the first developed newly improved varieties were BG1111, right? BG1111, BG35, and BG38, right? So if you take BG1111, that's a 4 to 4.5 month variety. And then these two varieties are 5 to 6 month age class, right? Age class. So the first newly improved variety, if someone asks in the oral exam or something, anywhere you go, right? Okay, can you hear me? There was an interruption.
Yes, sir.
Okay.
Yes, sir.
Okay, so the first newly improved variety is the BG1111, right? So this is how you write it, right? Then, right? So you have to write like this, BG1111, right? Was the first newly improved variety released by Sri Lankan breeders in the early 1970s, right? 1970s. So the parents, you should know this. This is because these two were not performing well, right? So we have imported something from Indonesia, right? So that variety called as Enkatek, right? To develop this BG11. But you should know in the in later development, right? In the BG1111 was the first newly improved varieties, but you now know, right? There are so many. In the next class, I will provide the list of newly improved varieties with their duration, especially. We had so many newly improved varieties now, right? So for to develop those newly improved varieties, later they used this TN1 and IR8 in the breeding program. There was another one called IR162, something like that. So they used so many varieties from the International Rice Research Institute because the collaboration increased. So our Rice Research and Development Institute had high level of collaboration with the International Rice Research Institute in Philippines. So most of the late improved varieties, the parents were received from the Philippines, right? Philippines. But the first, this NIV, that's the BG1111, had the parent from Indonesia, right? Had the parent from Indonesia. Okay, so in 1972, right, the Department of Agriculture decided to improve short duration varieties, because still you see this, the initially developed newly improved varieties had duration of more than 4 months, right? So this CBG 1111 4 to 4.5 months, the BG 35, BG 38, 5 to 6 month duration. Okay. So they wanted to develop short duration varieties, because to enable mostly cultivate Maha and Jala season, or maybe 3 seasons, right? If water is available, 3 seasons. So let's reduce the duration, they thought, right? So in that, with that thinking, right, the variety is BG 300. So that's the 3-month variety. BG 352, right, 3.5 month variety, were released in early 1990s, right? So if you remember this one, BG 300 was developed released in 1987. 1987, right? It's now 35 years ago, right? So these varieties BG 300 and BG 350 were released in the early 1990s to fulfill this requirement, right? Because the short duration, like 3 and 3.5, right? So these two varieties, right, are grown like more than 50% of annual cultivation extent even today, right? Even today, these are the two varieties very popular, right, among farmers, and cultivated more than 50% of the annual extent in Sri Lanka, right? So these are the two varieties that occupy the rice lands. Right. So, are there any questions in the development of newly improved varieties? Can you see the screen or no? Students?
Yes, sir.
Slides are moving, right?
We can't see the last slide which you have explained.
Okay, let me select. Okay. Okay, so what I was explaining is that since you couldn't see the slides, right? So they thought, right, earlier this developed newly improved varieties were, most of them were more than 4 months old, right? So they wanted to reduce the duration, so in a way that they can cultivate 2 seasons, right? So they, wanted to improve the short duration varieties of 3.5 months, right? So in, with the thinking of this, problem, right, they developed BG 300 and BG 352 and released in early 1990s. BG 300 was the first short duration variety released in 1987, and then BG 350, right? So then these varieties even now they are grown, right, popularly because of their short duration, more than 50% of the annual extent in Sri Lanka, right? So this figure, right, I wanted to show you, right, the distribution of different rice varieties, right? Rice varieties. So in the x-axis, right, if you see here, right, in the x-axis we have the year, right? So that starting from 1950 to its end somewhere in 2009, right? 2009. Still not very much updated graph, but still you can see, right? By 2009 we had so many newly improved varieties, right? Newly improved varieties. So starting from 1950 to 2009, right? In the y-axis you have the percentage of extent, right? Extent cultivated, right? So different lines shows different varieties. For example, this blue with triangle, right? This one is the traditional variety, right? And then old improved varieties by this purple line, right? And then the newly improved variety is dark blue, but the square type, right? Square type graph, right? So what you should know is that, right? Because of this improvement, right? Majority of them right now, in the late, here somewhere here, right? In the late 1960s, right, or maybe early 1970s. So this NIVs, right? NIVs started, right, to develop, right? So this extent is continuously, you see, right, continuously increasing and see it's almost around 98%. Now we cultivate our newly improved varieties, right? Newly improved varieties. So earlier we had, so if you go, but on the other hand, if you go to the newly improved varieties, traditional variety, right? Up to this, up to the development of old improved varieties, mostly we had like 100% traditional variety, right? Traditional variety, but it went down, right? Going down, and then now almost very less, right? Maybe 1%, something like that. We grow, right? Traditional variety. See here, this line. This is the line, right, of traditional variety, right? But if you take the old improved variety, right, that improvement occurred started in late 1950s, right? So they increased the extent, at least they were maximum around like 1970, right? Before these newly improved varieties started, right? And then they also went down, right? Went down. Because now majority of them we have is the newly improved varieties. Okay. Are there any questions? So the reason for this, the high rate of adoption of newly improved varieties is they are beneficial characters. Like they are high yielding, they are short type, they don't go for lodging, right? They are resistant to pest and diseases, they are resistant to several abiotic stresses, right? Sensitive, right? With all those beneficial characters, right? Now our rice growing systems replaced almost with the newly improved variety, right? But still in some areas, depending on some varieties, some traditional varieties are preferred by the people in that region. So they still grow, like especially in the northern region, right? Some of the varieties, not improved varieties, are grown because they prefer like some of them, like red pericarp and all, right? But when you look as a percentage of extent, right, cultivated in Sri Lanka, that is very less, right? Any questions before we wind up? someone asked earlier also, do you need to remember these values in the graph and all? No, right? But you should know the pattern, right? Pattern, and you should able to maybe tell when you write the answers. Somewhere in the 1970s, the newly improved varieties were developed. Then this improvement happened. Now, right, almost we grow newly improved varieties. So that kind of message is needed, but the values are not required, right? You don't need to just copy the, memorize and copy the exact graph, right? But this graph is very useful. You can in one picture you can see the milestone of rice improvement, right? Because traditional variety, the extent of cultivation went down. Then when they started to fall, right? When they start working on old improved varieties, right? Old improved varieties. So then our old improved varieties were picking up, but the newly improved variety technology emerges, right? Here somewhere, right? Then it's falling down, right? But finally, everything was picked up by the newly improved variety because of their beneficial characters. So that's the kind of milestone it shows. So if you don't have questions, so we will stop here, and then I will meet you tomorrow at 9. Thank you.